learners. Myself Dr. Sandeep Kumar Bansal, Professor, Ramesh Institute of Vocational and Technical Education, is discussing the unit 2nd of Pharmaceutical Organic Chemistry 2nd, that is BP 401T. This unit consists of geometrical isomerism, conformational isomerism, atrope isomerism and stereoselectivity and stereospecificity. Now, in this present lecture, we are going to discuss about the stereoselectivity and stereospecificity. So, these two words looks very similar. So, now we have to understand these two terms very clearly, means if these two terms are similar or these two terms are different, means how they are similar to each other, how they are differ to each other. Now, first of all, the question arises is why stereoselectivity and stereospecificity, means what is the need of stereoselectivity and stereospecificity. So, if we look upon the challenges or the problems in organic synthesis. So, major objective of synthetic chemist, organic chemist is to synthesize an organic compound containing multiple chiral centers as in a single specific stereoisomers. So, if any product contains number of chiral centers, so there will be number of configurations possible for that particular compound. Now, as more as the conformations are possible, the challenge is more. So, the challenge lies that we have to be very specific in synthesizing the product. If we want to synthesize R isomer, should it be R? If we want to synthesize, if having multiple RR or RS, whatever configuration is required. So, to synthesize that configuration is very important. So, it is also desirable that stereochemical transformation be carried out in predictable manner, so that the synthesis can be used to establish configuration of the molecule. Means sometimes synthesis, as we have discussed in lecture, that the methods to determine the configuration of geometrical isomers. So, similarly, if we are using any particular method in the configuration of a molecule, in establishment of the configuration of a molecule, so it should be very predictable, it should be very clear cut. So, that one should not have any mistake, one should not commit any mistake. Now, the stereochemistry at chiral center, there are two controls basically. If you see, there are two controls, the stereochemistry at a chiral center during a reaction in a fixed, either by conducting reaction under kinetic control or thermodynamic control. Now, what does it mean? So, let me explain these two words, why we are considering these two terminology, kinetic control or thermodynamic control. So, the stereochemistry at a chiral center during a reaction in fixed, either by conducting the reaction under kinetic control, in which the stereoselectivity depends on the differences in the free energy of the respective transition state. So, if reaction is under kinetic control, so the stereochemistry, stereoselectivity of the product is depending on the transition state, like SN2 reaction. So, in SN2 reaction, the reactivity is stereoselectivity is depending on the transition state. But if we talk about thermodynamic control, in which the stereoselectivity depends on difference in the free energy of the product. So, reactions, stereoselective reaction or stereospecific reaction are under the kinetic control or thermodynamic control. So, these are the specific parameters. So, these are the challenges ki why we should discuss, why we should know about the stereoselectivity, why we should know about the stereospecificity. So, to be selective to get the specific desired products or to get confirmation clear, to get the configuration clarity, predictable manner. So, one should understand, we should know why stereoselectivity and stereospecificity are required. Now, let us see what is stereospecificity. If we break this word into its reaction is stereospecific. This reaction is stereospecific. Now, what does it mean? Stereospecificity is the property of the reaction mechanism, where starting material differing only in their configuration are converted into stereoisomeric product. Means it is totally depending on the reactant or the reaction mechanism. Say for example, here is the I have quoted the example of SN2 reaction. So, reactants are deciding, they are pre-deciding factors, predominating factor, that they are deciding the stereochemistry of the product. Thus, a stereospecific process is necessarily stereoselective, but all stereoselective processes are stereospecific. So, we have to be very clear, we have to be very specific about the differences between these two words, stereospecific and stereoselective. So, stereospecific may be total 100% or partial. So, this term is also applied to the situation where reaction can be performed with only one stereoisomer. So, let us have an idea about this stereospecificity and stereoselectivity. How is it? Stereoselectivity means stereoselectivity is the property of the reactant mixture, where a known stereospecific mechanism allows the formation of multiple product, but where one or subset of the product is favored by factors such as steric axis that are independent of the mechanism. So, stereoselectivity is of great importance in the design of synthesis of natural products. So, these are the typical definitions of stereospecificity and stereoselectivity. So, let us try to understand them with few examples, but before proceeding with examples, let me differentiate between these two terms. So, definition wise, if we see, stereospecific reaction is a reaction in which stereochemistry of the reactant molecule completely determine the stereochemistry of the product without any other option. Remember, stereochemistry of the reactant completely determines the stereochemistry of the product without any other option. There is no option. Means reactant is entirely deciding or entirely responsible for the stereochemistry of the product. But if we say stereoselective reactions, so stereoselective reaction is a reaction which there is choice of pathway, but the product stereoisomers formed due to its reaction pathway being more favorable than other variable. Means one stereoisomer is favored over the other. This is very specific. So, these definitions will be more and more clear when we discuss about the more examples. So, number of products, how many products are getting prepared in this stereospecific reaction? Stereospecific reaction give a specific product from specific reactant, but in case of stereoselective reaction, there can be multiple products. So, in stereospecific reaction, there are chances of getting one product from selectively from one reactant, but in stereoselective reaction, the products can be multiple and one product can be favored over the another. Effect: stereochemical information of the reactant is preserved. This is very important, but stereochemical information of the reactant is lost. So, these are the three major differences between stereospecific reactions and stereoselective reactions. Now, let us elaborate stereospecific reaction with the help of one example So, as I have told earlier that stereospecific reaction is one in which when carried out with stereoisomeric starting material gives the product from one reactant, that is stereoisomer of the product from the other. Let us see what is happening in this. The reactant is R and we are carrying out SN2 reaction in the presence of sodium hydroxide. So, it is iodine is substituted with hydroxyl group and product, the configuration of product is S. On the other hand, if reactant is S, product is R. That means there is inversion. If we are using reactant R, product is getting S. If reactant is S, we are getting product R. So, this definition tells that gives the product from one reactant, that is stereoisomer of the product from the another. So, this is very important line and this line is very much clear with this example, means R is giving S and S is giving R. So, both the products what we are getting each time, they are stereoisomer of each other. So, this type of reactions counts for stereospecific reactions. So, stereospecific relates to the mechanism of a reaction. The best known example is the SN2 reaction, which always proceed with the inversion of stereochemistry at the reacting center. So, I think you must have got clear idea that what a stereospecific reaction means. Let us see what mechanism is favoring this stereospecific reaction. If we talk about SN2 reaction, SN2 reaction substitution takes place, nucleophilic substitution takes place from backward side. So, leaving group is leaving from front and nucleophile is coming from the backward side. So, this change in configuration means leaving group is leaving from front and nucleophile is coming from backward. So, there is a leading to the inversion and this inversion is stereospecific. Similarly, there is one more example. See, this is the elimination, beta elimination example. Again, if we see this methyl and hydrogen atoms are at variable position. Methyl is represented by wedge bond and hydrogen is a dash bond, means one is out of the plane towards us and one is from away the plane. So, in this case we are getting E alkene and when CH3 is away and H is towards us, then we are getting Z alkene. So, it is very clear that a specific reactant will give a specific product. So, if this type of reaction is taking place, so we are calling that reaction stereospecific reaction. So, a reaction in which stereochemistry of the reactant completely determine the stereochemistry of product without any other option. So, I am highlighting, I am asking the same statement again and again. So, what is stereospecific reaction? Stereospecific reaction is in which the stereochemistry of the reactant completely determine the stereochemistry of the product without any other option. One more example, addition of carbene to cis butene and trans butene specifically gives cis 1,2 dimethyl cyclopropane and trans 1,2 dimethyl cyclopropane respectively. So, these examples are enough. This is cis, we are getting cis, it is trans, we are getting trans. So, there is a specificity, remember you will never commit mistake, specificity with reference to reactant. So, products are specific to reactant, means what reactant we are using, it is only the determining criteria, it is only the determining which product we are going to obtain. So, reaction is very specific. Stereospecific again means that reactant is determining the specificity of the product. So, if this type of reactions are taking place, so we are calling them stereospecific reactions. Now, let us see stereoselective reaction. Selective means there is selection. There is a selection of what? So, stereoselective process is one in which one stereoisomer predominate over another. Means here we are having two products C and D from the reactant A and B. So, we have to select one, and if this type of situation is arising, then surely we are calling the reaction stereoselective. Remember, if situation is specific and selective, if product is reactant specific, we are calling stereospecific. If we have to select one product from the mixture of the product which is dominating, then the reaction is surely is stereoselective. If more than one reaction could occur between a set of reactant under the same conditions giving product that are stereoisomers, and if one product forms is greater amount than other, the overall reaction is said to be stereoselective. So, we have to focus on the very clearly on the word selective and specific. If product is reaction specific, then we are calling it stereospecific, and if product we have to select predominating product from the mixture of the product, then we are calling it the stereoselective reaction. In this case, we can say that C is 75% and D is 25%. So, C is predominating over the D. So, stereoselective reactions yield predominantly one stereoisomer of one pair of enantiomer of the several stereomers. So, it can also be defined as a reaction in which there is a choice of pathway, but stereoisomeric product is formed due to its reaction pathway being more favorable than other available. Let us see what does it mean. So, let us understand this with this example. In this case, OH is represented by the Wes bond, and we are carrying out elimination with the sulfuric acid. So, we are getting here again mixture of product trans and cis alkanes, and definitely surely one would predominate over another, and if this type of situation is occurring, then the reaction is we are calling stereoselective reaction. Similarly, there is one more reaction, the elimination reaction of beta iodobutane. In the presence of tertiary potassium butoxide using the solvent DMSO. So, HI is getting eliminated. In this case, we are getting mixture of three products, namely 2, 3 and 4. I have numbered them to discuss them easy. So, products we are getting trans 2 butene, that is 2, cis 2 butene, that is 3, and 1 butene, that is 4. But among these 3, 2 is maximum. So, whenever there is choice, there is selectivity, then the reaction surely is called stereoselectivity, and whenever the reaction is specific to reactant, product is specific to reactant, then definitely we are calling the reaction as stereospecific. So, using these criteria, we can easily define or we can easily differentiate between stereospecific and stereoselective reactions. Now, let us see what are the basics of stereoselectivity. Now, for stereoselectivity, the substrate must be pro-stereogenic. It is as like as prodrug. So, product reactant must be, substrate must be stereogenic, which in turn depend on the symmetry or more specifically on the topology of the reacting group or phases of the appropriate modification give rise to stereoisomers, enantiomers or diastereomers. So, stereoselective reactions can be easily differentiated or can be categorized into diastereoselective, enantioselective and regioselective. So, the principle of stereoselectivity, whether it refers to diastereoselective or enantioselective, is a kinetically controlled. So, by the virtue of their diastereomers nature, they would differ in the free energy and thus give product in different amount and the great difference in the energy, here is the stereoselectivity. So, here is the kinetics or the energy of the reaction that is eventually controlling the product, difference in the stereoselectivity. If the difference is of the order of 10 kilojoule per mole at ambient temperature, so definitely one product will predominate over another more than 98%. So, students, remember this kinetics is totally controlling the reactivity, stereoselectivity, which product it, whether one product will dominate, how much will be the ratio, it will be 60-40, 70-30, 80-20, 90-10 or whatever is the ratio. So, it will be determined by the energy difference. If it is more than 10, then surely the product will be more than 98% yield. Now, I have told that we can divide or describe the stereoselective reactions in diastereoselective, anisoselective and regioselective. So, stereoselective reaction in which possible products are diastereomers is said to be diastereoselective. Let us take this example 1,2-dimethyl cyclohexane, we are getting 1s2r, 1r2r. So, this is SR combination, RR combination. So, both are diastereomers to each other. So, if this type of case arise, we will consider this reaction as a diastereoselective. So, there is one more example. From this product we are getting cis 62%, trans 32%. So, this they are not enantiomers, they are rather they are diastereomers. So, therefore, whenever any stereoselective reaction which gives the product that are diastereomers, the reaction is called diastereoselective. If similarly, if a reaction give product that are enantiomers of each other, the reaction would be considered as enantioselective. So, using this example 4-butyl 1-methyl cyclopentane 1,3-dione. So, we are getting one product that is S in nature, another product that is R in nature. So, if we are getting these type of products, so one can say that reaction is enantioselective. If we are getting constitutional isomers from a particular substrate, mixture of regioselective, mixture of constitutional isomers, then we are calling the reaction as a regioselective. So, this is one more example. So, reaction stereoselective reaction can be enantioselective, it can be diastereoselective or it can be regioselective. So, here is a summary of the stereospecific reaction versus stereoselective reaction. So, stereochemistry of the reactant determine the stereochemistry of the product. It is a very clear cut difference, and one stereoisomer is favored over the another in case of the stereoselective reaction. Here are the examples are shown that alkene is E and Z in the stereospecific reaction. Here we are getting again alkene, but one alkene is predominating over the another. So, it is the example, perfect example of stereoselective reaction, and stereochemical information of the reactant is preserved in this case, and stereochemical information of the reactant is lost in this case. So, these three differences are the marker differences that are between the stereospecific and stereoselective reactions. So, now here are the certain definitions from some standard books like Alien. So, I am quoting here stereoselectivity, the preferential formation of one stereoisomer over another in a chemical reaction. If stereoisomer is enantiomer, one speaks stereo of enantioselectivity. Similarly, stereospecificity is also there. So, these two definitions are sufficient. These are the standard definition from Alien book. Similarly, here are the two more definitions from Ansel and Doctrine book. The stereoselectivity how it is defined and how stereospecificity is defined. Now, how to assign, how to differentiate, how to understand which reaction is stereoselective and which reaction is stereospecific. So, we have to look upon the reaction if we are getting multiple products and one isomer is predominating on another, surely the reaction is stereoselective, but if we are looking for one stereospecific product, then the reaction is stereospecific. So, let us have one example. So, this initially this reaction looks stereospecific, but no, this reaction is stereospecific. So, you have to think twice, thrice, number of times that why this reaction is stereoselective rather than stereospecific. Similarly, there are two more examples that you have to focus, you have to be very clear in these two terms in deciding whether these reactions are stereoselective or stereospecific. So, in summary, stereoselectivity has been classified two categories, enhancer selectivity and stereoselectivity, and one more here we can add, that regioselectivity. Stereospecific reactions are reactions in which stereochemically different molecules react in a different way to give cis and trans stereoselective reaction. So, my dear students, I hope you must understand the difference between stereoselectivity and stereospecificity. So, this was the last topic to be discussed under this unit second. So, thank you very much. Thank you all.